Welcome everyone. Thanks for joining us today. Before I introduce today's speaker, I just want to remind you all that we are always welcoming suggestions for food and speakers or topics. So please drop us an email if you have any suggestions at foodforsoot@food.gov.uk. Today's talk is from Dr. Craig Baker-Austin, who is Principal Microbiologist in the Food Safety Group at the Center for Environment, Fisheries and Aquaculture Science, CFAS. Craig leads the scientific research work associated with various aspects of bacteriology, but with a focus on established as well as emerging pathogens of human health relevance. His primary research experience and background is interdisciplinary and bridges the gaps between microbiology, risk assessment, molecular biology, and waterborne, foodborne disease emergence. Craig has over 20 years experience in the field of molecular and public health microbiology, developed during his PhD and postdoctoral positions, and more recently as a senior microbiologist at CFAS. He is recognized by the FAO of the United Nations and the WHO as an international expert on waterborne and foodborne pathogens. He leads an internal method working group on pathogenic vibrios, ISO WG27, and he is an expert member of Vibrio risk assessment at EFSA and currently holds an honorary lectureship position at the University of East Anglia and an honorary professorship at the University of Exeter. We are very pleased that he has accepted to come and speak to us about aquatic zoonotic pathogens and about epidemiological and demographic factors that make their study critical. Right before I hand over to Craig, I will just remind you of the housekeeping rules. Since we are on Teams Live, you will not be able to unmute your microphones to ask questions, but you will be able to post these in a Q&A. The chat will be right on your screen. Please feel free to post your questions whenever you like, but do note that we will cover these only at the end of the talk when I will read them out for Craig to answer. Finally, please note that this seminar is being recorded. So now, over to Craig. Thank you.
Thank you very much. It's a real pleasure to be here today and to present for you. So this presentation is really rather broad, and it's a whistle-stop tour of some of the things that are, I would imagine, or that I think are important from a foodborne perspective. And I'm focusing really on zoonotic pathogens as an emerging foodborne risk. So as an outline, I'm going to talk briefly about what are zoonotic pathogens. Then I'm going to focus really on microbial risks associated with seafood, which is what I work on. I'm going to talk a little bit about some of those specific risks. Vibrios will be one of them. I'll talk a little bit about emerging viruses in seafood, covering things like hepatitis E virus and others. I'll then show you some case studies from around the world, how we're now better able to understand and control risks. And really the reason why we can do that is using cutting edge science. And then I'm going to focus a little bit towards the end on what the current risks are in the UK. So what are zoonotic pathogens? So these are infections that are caused by diseases that are spread between animals and people. The severity of these varies very much so from mild to life-threatening. Now, zoonotic foodborne pathogens are caused by the consumption of food or water contaminated by pathogen disease-causing microorganisms such as bacteria, viruses and parasites. So they enter the body through the gastrointestinal tract, where the first symptoms typically occur. The most common foodborne illnesses are caused by bacteria such as Campylobacter, Salmonella, Yersinia, E. coli and Listeria. And these are not a trivial cause of disease. So foodborne zoonotics are a significant widespread public health threat. In the European Union, for instance, there's over 350,000 reported cases each year. It's probably much higher than that. That's based on the available epidemiology. And there's a lot of factors that suggest that the number of cases are actually a lot higher than that. So the focus of this presentation is on new and emerging zoonotic risks from marine and aquatic environments. And I'm going to try and place a particular focus, a particular emphasis on food. So you're probably asking what the microbial risks are in the marine environment, and they're actually very varied. There's a wide variety of pathogen risks that are present in marine waters. For example, fecal contamination, you hear about this all the time at the moment in the news. Sewage contamination, it introduces a range of different pathogens into the aquatic and marine environment, and that includes bacterial, viral, protozoan agents So these introduced agents include viruses, so enteroviruses, poliovirus, hepatitis A, norovirus and rotavirus. There's also some bacterial pathogens that are found in sewage as well. That includes salmonella, campylobacter, shigella, and also not as common, but Vibrio, cholerae 010139, as well as E. coli. Now, those are what we call introduced pathogens. There's also naturally occurring pathogens present in marine and aquatic environments, and those include Vibrio species and also harmful algal biotoxin producing organisms. And these are widespread in marine waters around the world. So we've known for a long time that water and shellfish are an effective vehicle in disease transmission. So you can get infected directly from drinking water that's contaminated with a range of different bacteria, protozoa, viruses, etc. But also bivalve filter feeding shellfish, in particular oysters, clams, mussels, very, very effective at concentrating whatever's present in the overlying waters, and those can then cause infections if you consume those shellfish species. Certain pathogens, one of those would be Vibrio cholerae, can then also cause widespread community infection. So you get an infection yourself from water, and then it can be spread to others quite effectively. Another example of that is typhoid, and there's a wide variety of different pathogens that are present that can be transmitted in that way. And we've known about this really for about 200 years now. The first outbreaks associated with shellfish were reported in medical journals at the start of the 19th century. Now, because these organisms, mussels, clams, oysters, very effective at filtering whatever's present in the water column, they can actually concentrate certain bacteria 100 times higher than in the overlying waters. And there's been some studies that show that oysters and mussels, for instance, have got the capacity to filter water at an incredible rate. It's equivalent to a human being able to consume an Olympic-sized swimming pool per hour. So that's their kind of filtering capacity. And the outbreaks associated with shellfish are often really, they can be not trivial at all. They can be really significant. We had a hepatitis A outbreak in China in the early 1990s. There was 300,000 reported cases, and that remains to date one of the largest foodborne outbreaks that's ever been reported worldwide. Now, the risks associated with shellfish have actually changed quite considerably over the last 100 years or so. So one of the predominant risks of consuming raw bivalve shellfish used to be typhoid fever. That's an important zoonotic pathogen. It's caused by the bacterium Salmonella cerevisiae typhi. It's frequently a deadly disease. And actually, most of the microbiological controls that are currently in place, not just in the UK or in Europe and internationally, were driven by the need to control this particular pathogen. Now, typhoid is no longer an endemic disease in the developed West, but we do have other pathogens as well. Norovirus is the most significant pathogen associated with raw shellfish consumption. Now, I'm going to talk about some different types of emerging risks, and the first one I'm going to mention is Vibrios. So these are common natural constituents of the marine environment. There's about 100 species of Vibrios that have been reported to date, and of those, about 12 can cause infections in humans, and they're responsible for a wide array of shellfish and waterborne associated diseases. What's really important about Vibrios is that they are extremely sensitive to climate change, and we're now starting to see outbreaks in environments where these bacteria have traditionally not been endemic or not been found before. Certain strains, certain species also have pandemic potential, and they're capable of rapid pandemic spread, so they can move around the world very, very quickly. Several species of Vibrio are zoonotic. By their very nature, they're capable of infecting humans and marine animals. And also, one of the things that I'll mention a little bit more detail later on is that we have changing demographics. So there's various factors, including an aging population, more people with underlying risk conditions, that will make these pathogens a severe problem in the future. This image that you can see in the bottom right is this is a necrotizing wound infection. This is from a man that was in a fish market in Japan, put his hand into a basket of shrimp, cut the back of his hand, and you can see the incredible amount of tissue damage that was caused here. And this, unfortunately, this individual actually died, so they can cause very severe infections in humans. So there's two main routes of infection from Vibrios. The first is through the consumption of raw or undercooked seafood. The real at-risk food items are bivalves that are consumed raw or lightly cooked. So that's really mostly oysters, but also clams and mussels as well. There's also been a big increase in recreational exposure. So if you go swimming in the sea with cuts, wounds or abrasions, they come into contact with water that's got Vibrios in them, and you can get a wound infection that way. And those can actually progress to more serious complications, such as primary septicemia. Now, whenever I do a presentation and I mention Vibrios and climate change, I always show this figure here because I think it's a nice figure from a science paper. It paints a thousand words. And this is actually from a study site in North Carolina, and it was looking at the prevalence of Vibrios in the water column in response to ambient temperatures and through a few different seasons. And as you can see, the Vibrios and temperature track each other almost perfectly. So you get an increase during the spring and summer, and they become far more common. And then during the winter, they die off. We believe they end up going into the sediments, and then they go into a viable but non-culturable state. And then when the water temperatures increase again, they start to replicate and proliferate again in the water column. So there's a very clear relationship between temperature and the abundance of these bacteria, not just in the environment, but also in the diseases that they can cause in humans. Now, probably the most important seafood associated infection from the Vibrio family is Vibrio parahaemolyticus. I'll just call that VP from now on. It's common, naturally occurring. It can infect various aquatic species as well, such as shrimp, so it's a zoonotic. It concentrates very readily in bivalve shellfish, such as oysters and clams, and it's the most common bacterial infection associated with the consumption of seafood. And it's often associated with very large foodborne outbreaks. We do now find this pathogen in UK shellfish and also in water. We find this regularly during the summer months. What's really interesting about this particular pathogen, this particular species, is that it is capable of pandemic spread. And in 2012, a new variant of Vibrio parahaemolyticus emerged. It was a strain that we'd only ever seen in the Pacific Northwest region of the United States, and then it turned up on the east coast of the United States, so 3,000 miles away, and also in Europe at almost exactly the same time. This particular strain, we call it PMW because it stands for Pacific Northwest, is a really huge concern. It's caused major shutdowns of the the US shellfish industry in 2012 and 2013. And the ability of a particular strain, a particular species to spread very quickly, pandemic spread, is a major global food safety and economic issue. So it's something that we need to keep an eye on, specifically as a food testing laboratory. So what's happened in the last sort of 2 or 3 decades is that Vibrio parahaemolyticus has spread all around the world. And what's interesting, actually, is that it's spread from the same place that we tend to find cholera, which is the Bay of Bengal. Most of the pandemic waves of cholera have emerged in that region and then moved around the world over different time periods. And interestingly, parahaemolyticus has done exactly the same thing. We don't quite understand why that is. We don't, but it's something that has happened now on several different occasions with both of these different species. What happened in the late 1990s, a new strain emerged called O3K6, highly pathogenic. It emerged in this region here, and then it spread all to the continent on Earth very, very quickly. And a few years later, in 2012, we had a new variant. This is the one I mentioned, the Pacific Northwest strains. Those emerged and moved to the eastern seaboard of the United States and then into Spain at exactly the same time in August 2012. What's really frightening, actually, is that the Pacific Northwest strains are highly, highly pathogenic. So this figure was generated, I believe, using a very limited volunteer study from the United States. And the black lines you can see on the right hand side are data from a couple of different sources, but they show the normal pathogenic strain, so that's the O3K6, and then on the left hand side the red dashed line is the Pacific Northwest strains. So to put this in some context, you can eat an oyster with 100,000 to 1 million strains of Vibrio parahaemolyticus, and it would probably initiate an infection from the K03, K6 strains. But if it was contaminated with the Pacific Northwest strain, you're probably looking at maybe 3 or even 4 orders of magnitude lower a dose to cause and to initiate a human infection. So you need far fewer of these cells to cause an infection in humans, and this is why we're really worried about this particular strain, because if it moves around quickly, we will probably see lots of foodborne incidents associated with this particular serovirus, this particular strain. Now I'm going to move on to one of the other pathogens that I work on. Again, it's a zoonotic, Vibrio vulnificus. It's similar in many respects to parahaemolyticus. Again, it likes warm, low salinity marine waters. Typically, you find it when water temperatures get above 15 degrees Celsius. We do find this bacterium rarely in the UK. It is present, but very much at low numbers. It's quite a difficult bacterium to isolate from shellfish. It can occur in high numbers in seafood, such as bivalves, particularly again during the warmer months. This is a really important opportunistic pathogen, and it causes both wound infections and sepsis, again via exposure to seawater and through the consumption of seafood. Why this is an important bacterium, why we need to be focusing on this, is that it carries the highest case fatality rate of any foodborne pathogen. So infections mostly involve males, about 90% of cases, so it's a really interesting organism just from a demographic perspective. It also tends to infect the elderly and at-risk persons. We're starting to see now an increase in Europe and USA, and the infections are regularly reported in Asia as well. So to give you some idea what the sort of risk factors associated with this bacterium are, it's normally men, normally 45 to 60 years old. It can cause infections in humans and eels. The fascinating thing here is around 85 to 90% of cases occur in males, about 15% in females. There's around 100 cases a year in the USA, so it's quite a rare infection. You know, you're looking at a country that's got 300 million people, 320 million people, but the cases do appear to be increasing. We don't seem to see many foodborne cases in Europe. We do in the States. Perhaps half of those 100 cases each year are associated with consumption of seafood. Now, if you get a wound infection, it's got about an 18% rate. Now, this figure on the right-hand side, it's a little bit gory, but this is a gentleman from the Gulf in the United States. He had an insect bite on his ankle. He went in the sea and he got a Vibrio vulnificus infection. You can see the massive tissue damage that's occurring here and the spread of this infection. So these two pictures were taken only 4 hours apart, and about 24 hours later, unfortunately, this gentleman died. So it's a really severe human infection. It's also one of the most expensive infections to treat. If you get a wound infection, you're going to be hospitalized. You're going to be on antibiotics. Often, particularly in the States where we've got good data, you're going to be in hospital for quite a long time being treated for this particular infection, and it often progresses to the point where you have to have tissue debridement or even limb amputation. So really not something you want to get, but they are very rare infections, but they're notable infections. And to put it in some context, this is a little table I put together of the most pathogenic microbes in the world, the ones you would have heard of: Ebola, Marburg, Crimean Congo hemorrhagic fever, Lassa fever, bubonic plague. It's in that sort of ballpark in terms of the case fatality rate if you get a foodborne infection, and even a wound infection. It's almost a 1 in 5 chance that you won't survive. So something we certainly need to keep an eye on for the future. I'm going to move away from Vibrios just for a second and talk about some of the other things that can that are on the radar, some of the other things that we're looking at. So hepatitis E virus, it's an important and emerging zoonotic pathogen worldwide. There's a lot of infections. There's 20 million human infections reported in 2005, 44,000 associated deaths attributed to hepatitis E virus in 2015. We've seen a big increase in hepatitis E infections, certainly in the UK in the last decade. The virus spreads from fecal oral route transmission, so it's shed in feces and urine. Infection with the virus usually occurs through the consumption of fecally contaminated water or undercooked or raw meat products from infected animals. Pigs are a big source of hepatitis E virus. Other transmission routes have been identified and theorized, but they remain quite elusive. Several studies have found hepatitis E virus in live bivalve shellfish, so there is a potential route for human transmission. In terms of what this looks like, where you can get these infections from the environment, we have a natural reservoir of hepatitis E virus, particularly in pigs and in deer and boar. You can consume those products. There could be runoff from those animals that you can come in contact with. There's also several routes through where the aquatic environment could contribute to the human transmission of these viruses. So irrigation water, shellfish and drinking water, and of course you're coming into direct contact then with the virus. And we don't yet know how important these different routes are, and it's something that we've been very interested in for a long time. And I co-supervised a PhD student that's just recently finished looking at the role of water and shellfish and hepatitis E virus, called Zoonotic. Something else that I'm going to mention very briefly is COVID. That's also shed in feces and urine by infected individuals. And various studies, you probably would have seen them over the last few years, have identified using quantitative real-time PCR COVID-19 in wastewater, and that was actually used as a means of being able to see where hotspots of disease transmission were taking place in the community. At the moment, there's no current evidence of transmission of COVID-19 via a food vehicle. As far as I'm aware, that's still the case. There's no evidence to date on the presence of COVID-19 in bivalve shellfish. And encapsulated viruses like COVID-19, they tend to have a lower environmental stability. They tend to break down far quicker when they're released into the environment compared to, say, something like norovirus, which is a much hardier, tougher virus. However, all of this is based on quite limited data. And recent risk assessments suggest the risks from food are very low, but the levels of uncertainty with these risk assessments are quite high. So again, it's something that we need to keep an eye on in case it changes in the future. I'm going to talk now about how we've been applying different scientific areas, different approaches, different tools to study these emerging risks. And this is a whistle-stop tour. There's bits of microbiology and virology in there. There's some risk assessment. I'm going to talk about epidemiology and also genomics, but not in any real detail. So one of the most exciting things that I've ever been involved in, working with collaborators at NOAA, which is the National Atmospheric and Oceanographic Administration in the States, and also University of Barcelona, is using satellite imagery. It's using these weather satellites that we have a network of now. We've had for the last 30 or 40 years. And what they actually do is that they detect infrared radiation that's emitted from the Earth's surface, and you can use that to infer surface seawater temperatures and land temperatures as well. And there's several satellite systems that have been carrying out this work for a long time. And what these allow you to do is look at any region on the surface of Earth with a high degree of resolution. And what I'm going to show you now is a couple of case studies, one from the United States and one from Europe. And what these, I think, clearly show is that during and following outbreaks, you can actually use this kind of data sets, these kind of approaches, to look for environmental signatures that are associated with large-scale outbreaks. So the first one I'm going to show you is an outbreak from northwest Spain from 25 years ago now. So this is an epicurve at the top. This is the number of reported cases, and on the bottom you can see this body of warm water moving along the coast of Portugal into Vigo, where these infections were reported. This is a reasonable size outbreak. 35 cases of shellfish associated illness were reported during this time period. It's important to point out prior to this, and then afterwards there was no cases. So in both time and space, we've got this clear environmental signature of this movement of warm water into the area associated with a quite sizable foodborne outbreak. I think that's a good example. I'm going to show you one that I think is slightly better, and this is from the Pacific Northwest region of the United States, and you can see here this body of water moving into California, then in May, and then it moves up further up the coastline into British Columbia in July, and then in Washington state, and then finally a few weeks later into Oregon. And you can see where that body of movement of warm water has occurred. There's been a big outbreak. This was the largest, I think it still remains the largest Vibrio parahaemolyticus outbreak in the United States. 209 infections, all from seafood harvested from California, Oregon, Washington. One person died from this outbreak, and you can see the water temperatures are really notable. They're 3 to 5 degrees Celsius warmer than they normally are that time of year. And how we've developed these tools, because you can use satellites to gather temperature data, we've developed some risk mapping tools, and these also use salinity as well. And what we're actually doing now is providing a free web portal, so you can look at any surface on Earth. It's got a 7-day predictive function as well, but you can look to see where Vibrios are likely to be present in the environment. We call it a risk portal. I'm going to be careful using the term risk because it means different things to different people, but effectively it's the risk that these bacteria are going to be present in the water column rather than the risk that you're going to get an infection. There's some limitations to this kind of approach. It uses very coarse data, so 5 kilometer square sized grids for temperature retrieval. So it looks a little bit blocky in places, like a very old computer game, maybe from the 1980s or 80s or 90s. But we're improving this all the time using better resolution data. But this is free to use. So if you type Vibrio risk into Google, it's the first thing that comes up. I'm going to talk now about how the science has changed in the lab. So that's the satellite data that I've shown you there, and the risk prediction tools. But actually, there's been some big changes in laboratory testing over the last few decades. So it's really important when you get samples, foodborne samples, that you can isolate bacteria. And we've been doing that from the 1960s onwards, the development of better approaches to be able to isolate bacteria directly from food and clinical samples. From the 70s and 80s onwards, there was various improvements in biochemical tests. And then from the 90s, when I started working as a microbiologist, there's been a huge, an absolute shift in terms of what we're able to do. So molecular methods such as conventional PCR and later quantitative real-time PCR have come online, and they allow you to very quickly and readily isolate and identify and characterize strains of interest. And with quantitative real-time PCR, you can also get some idea about the loading. The quantitative element of there is really important to be able to see what's present in a sample. The most recent things on the far right hand side here are genome sequencing, and also MALDI-TOF, which is a very rapid means of being able to identify strains. It takes literally minutes. You can get a result in that's that quick. But what I've worked on, one of the things I think is most exciting, is next generation sequencing. So I'm sort of old enough now to remember doing sequencing like the old ways in the lab, having to break up an entire genome and sequence little bits of it that have been cloned, and that was very laborious, very expensive. It used to cost upwards of £1 million to sequence a microbial genome. Now you can sequence a bacterial genome, a standard size one, 3, 4, 5 million bases for £30 or so. So it's become a routine method, whereas before it wasn't. I'm going to give you an example of one of the things that we've been able to do. So this is using whole genome sequencing data from outbreak samples. And what we're actually able to do is, if you know when and where you got an outbreak, you can get the strains. Those can be sequenced and they can be directly compared to other strains as well at the same time. And then you can look at things like the emergence, the dispersal, and also the evolution of strains. You can also look into, some of the more practical things. How are these bacteria being transmitted? When did this outbreak take place? Can we pinpoint it with more accuracy? And it allows you to ask fundamental questions that you were not able to do even a few years ago. So you can apply this kind of approach for bacterial and viral pathogens, and this has only become available in the last decade. We started doing this work at CFAS in 2012, 2013, but in the last few years it's moved on very quickly. And what you can actually do, and what's being done at the moment with any pandemic, is those strains are very rapidly sequenced and identified. And to put that into some context, when we've had outbreaks in the past, it could take sometimes days or weeks to get results. Now you can do it in a matter of hours. So it's a really powerful set of tools, huge amount of granularity and resolution, and they've completely superseded all of the older methods that we used to use in the lab. So I'm going to mention some of the things that are going on at the moment in terms of risk, and why I think these types of risks are going to increase in the future. I mean, the first thing to mention is really we've got a rapidly aging population. So we've got far more people now that are susceptible to certain zoonotic pathogen risks from marine and aquatic environments. So that would be things like vibrios and also some of these other microbial agents as well. In particular, Vibrio vulnificus, Vibrio cholerae, non-O 10139. You're far more likely to get systemic and very aggressive infection if you're older. At the moment, I think it's about 1 in 10 people is age 65 or more. By 2030, it's going to be 1 in 6. So there's a huge increase internationally of people that are getting older that are going to be more susceptible to these types of infections. I suppose kind of linked to that as well, we have the issue of underlying conditions. We've got far more people now with underlying risk factors for zoonotic infections, in particular vibriosis, but some of the other things as well. Those include conditions like liver dysfunction, diabetes, compromised immune systems, etc. There's also exposure. It's important to think about that. There's a huge number of people that are living by and in direct contact with water. So there's a huge increase in potential risk exposure. I think something like in the United States, population of 320 million people, about half of that population live within 100 miles of the coast. So there's a lot of people that could potentially be exposed to these types of pathogens. And I think we also need to look at consumption as well. There's an increase in consumption of raw or undercooked shellfish. So there's also a greater international trade in shellfish commodities, and that will have ramifications with regards to risk. And we don't really know how that is going to bottom out yet. So how do we tackle risks? Well, for Vibrios, it's actually quite simple. These are very temperature sensitive bacteria. If you cool seafood, you greatly reduce human health risks. So this is some data on the right hand side from the United States. So what happened in 2012, we had that new strain that I mentioned earlier on, caused devastation along the eastern seaboard of the United States, and the NSSP, so that's the National Shellfish Sanitation Protocol, put together new rules which mandated that shellfish that were being harvested in those areas had to be iced, and that was a very, very effective means of reducing risk. And the number of cases, although it went up during 2012, 2013 with this new strain that caused these outbreaks, it's reduced down quite quickly afterwards. I think for other pathogen risks, things like hepatitis E virus, we need better environmental and baseline quantification data. We also need more effective surveillance and epidemiological data sets, and that's critical to identify when and where we have particular incidents or outbreaks, etc. Where things are moving at the moment, really rather exciting with the tools that are available to do certain tests to look for different emerging risks have improved rapidly. I mean, I showed you the example of the genomics, but we can now look for things like antibiotic resistant bacteria, in shellfish. We can do chemical residue analysis very quickly and easily using targeted and also untargeted approaches. We can look at the community profile in these sorts of samples, so you can see when and where certain pathogens are emerging. And you can also quantify things like antibiotic resistance genes, pathogen genes, virulence associated markers in these samples using a variety of different methods. And this has only been available really in the last few years. Where things are changing, though, is a sort of broader approach. We're far more, I think we're far more ready to embrace the One Health concept, which is not just looking at animals or humans or the environment separately and in silos, but it's looking at them actually as a larger entity with those interaction between those different elements. And actually, the science has moved on very rapidly in the last few years. The types of approaches that are available are really staggering. Where the difficulty lies now is actually analysing that data. You can take any kind of environmental sample. So this example here, using natural, rural, urban or industrial, you could take any kind of environmental matrix. Let's just take, for example, soil or wheat or shellfish. You can then test them for a variety of different things very rapidly using a huge array of different analytical tools. But where the difficulty comes in is how you actually crunch that data. What does that data mean? So that's really the challenge at the moment, being able to get all that information together and to be able to respond to it in a meaningful way. And that's really where things are moving to this operational One Health approach, where we're able to really fully understand what's going on across different levels of granularity, from molecular all the way up to landscape scales. In terms of sort of finishing off here, most of the emergent shellfish related pathogens that I described in this presentation have been found in the UK. I would say that actually, on the whole, we don't seem to see many reported outbreaks in the UK, but we need better systems of epidemiology reporting. We've got effective risk reduction strategies in place because we know that they work in other countries. So those could be used to tackle emergent risks. So the example I used there, icing shellfish, very, very effective, but there's also improved approaches for testing as well. And I think we need to have a coordinated and transboundary approach to sharing of data, especially epidemiological and surveillance data between different countries. So just as a quick summary, there's a wide variety of emerging zoonotic risks. There's a dire need for improved systems of epidemiology, surveillance and reporting, particularly with regard to these risks. I would say that, with demographics likely to increase risks in the future, there's also increased international trade of shellfish, so that's something we need to keep an eye on in terms of our intelligence. But we do have improved methods to tackle these challenges, and that's things like the remote sensing, so using satellite imagery to look for when and where we're likely to see outbreaks, but also the lab-based methods, detection and quantification of these pathogens, not just in food samples but also in clinical samples as well. And I'd also say that the science underpinning this work has really changed dramatically in the last decade. So thank you very much for listening to that. And just as a quick acknowledgement, a few people that I work with: Professor Jaime Martinez at the University of Barcelona, and Dr. Joaquim Tristains at the University of Santiago de Compostela in Spain. I'd also like to thank CFAS, CCORN for funding some of this work. Thank you.
Thank you so much, Craig. It was a very interesting presentation. I was really unfortunate and frustrated to lose connection in the middle of the presentation.
Oh,
must have been popping out on the screen. Hope I didn't disturb you. We've got a few questions that have come through already. A couple that came at the rather beginning of the talk. First one was, how would you define endemic? So very broad.
That's a good question. There's probably a very specific and universally accepted term for that, but it would be when a certain disease reaches a certain threshold in terms of its prevalence and whether it remains in the environment and in human populations as well. So that would be there's probably a far more universally accepted term for that, but those would be the key factors. Whether, for example, typhoid is not endemic in the UK anymore, but it was at some point in the past. So it's when those cases would have been reduced to a certain level, and when we don't see those infections reoccurring, that would be important to define that.
Thank you. Your slide talked about routes of infection, consumptions or cuts when in water, and someone asked whether you could get infected through bacteria getting into your nose, mouth, or eyes, for example.
Yeah, you can. So certain species tend to be associated with those types of infections. So Vibrio alginolyticus tends to cause a lot of ear infections and also eye infections, people swimming in the sea. But yeah, most of those, I sort of lumped all of the Vibrios in together, which perhaps should have been a little bit more careful there. But yeah, there's different strains that tend to be associated with different things. Alginolyticus is definitely the one that tends to cause a lot of those other sort of those other types of infections.
Thank you. Another question. You mentioned the level of bacteria is impacted by climate change. Do you think we will see an increase in cases or outbreak over the years as climate change increases? I was wondering myself about the impact of flooding, for example.
Yeah, I was sort of conscious of time, and I've done a huge amount of work in this area. But yeah, there's very clear evidence now that climate change is going to increase not just the prevalence, but also the geographical spread and the temporal characteristics of these pathogens. So something like a heat wave, we've got a lot of evidence that when we have heat waves, particularly in Northern Europe, people go swimming in the sea, we get a huge increase in Vibrio infections. Also extreme weather events such as storms. So following, for example, Hurricane Katrina, which made landfall in the United States in August 2005, there was a huge increase in reported Vibrio infections, and these were not food infections, these were wound infections. So people wading in flood water that have got cuts and grazes and abrasions. So those things are very, very important with regards to disease transmission.
Thank you. We've got a question on methodology there. Was LAMP, so I think it's loop-mediated isothermal amplification, also used in zoonotic pathogen testing? Yeah. This was used a lot for COVID, as I'm told.
Yeah, it's been used very, very readily for Vibrios, Vibrio parahaemolyticus and Vibrio vulnificus. There's several published assays for LAMP for those bacteria. It's got an advantage in that it's very quick. You get results very rapidly, and it's also quite, I think it's quite a sensitive and specific method. I actually don't have a huge amount of experience using LAMP, but I know that it is used for a lot of these bacteria.
On a more kind of maybe prevention strategy, what do you think is the biggest risk to human in terms of future potential pandemics? And do you have, or are there any prevention strategies for the public that are being developed?
That's a great question. Well, that is a very tricky one because you have to sort of have a crystal ball. It's tough actually because I'm a bacteriologist, and what we've had to go through the last 3 years has been driven by a kind of respiratory virus. So there's a lot of research in that area. There's a lot of concern about new flu variants. So in particular, bird flu. You probably read that a lot in the media. I don't know actually. From a bacterial perspective, I think antibiotic resistance is a massive big issue at the moment. So I'm moving it away from viruses back over to bacteria there. But if we don't have the drugs to be able to treat even basic common infections associated with bacteria, then we're going to be in serious trouble. And I think that's going to be one of the main challenges in the future, and I think it already is actually.
I think we definitely had that with strep A this year. Another question, what is the difference in the risk between consuming shellfish versus meat?
I think that, they have completely different risk profiles, and they're so broad that it's quite a difficult question to answer, but I'll attempt it. So I think we tend to see outbreaks linked to consumption of raw or undercooked seafood. And because shellfish are very effective at concentrating, in particular, bacteria from the water column, that's where you can get those risks, where you get a high loading of bacteria. Shellfish that are cooked, the risk profile is obviously a lot lower because it's going to be, well, pretty much microbially inert. Similarly, you can have an issue with meats that are prepared in a particular way have a different risk profile to meats that are, say, for example, fully cooked. So taking something like hepatitis E virus, for instance, that's found very commonly in sausages and in blood sausages. So that would be a riskier food commodity than, say, something that was fully cooked, like a normal sausage.
Yeah, so it's processing related, really. Do you know if governments are looking into these emerging risks to change legislation and industry requirements to tackle them?
I think certain pathogens are on the radar. So I think that Vibrios are, there's interest from the ECDC and EFSA for Vibrios at the moment. I don't know whether that will lead to legislation or not. It's difficult because we don't have in Europe, these are not notifiable pathogens with the exception of cholera, which I didn't really mention as part of this presentation. But I think if they became notifiable, that would be the first step with regards to future legislation. At the moment, they're not. So you could get a Vibrio vulnificus infection and it's not reported. In the States, those have been nationally notifiable since 2007. So if you get an infection in America, you have to report it through a state lab to the CDC. So that information, that data is gathered. And I think that's the first step, really, with regards to legislation.
That's interesting. So how do you gather information about their appearance here in Europe, for example?
Well, I mean, I've got a network of collaborators. I work as the director of the UK National Reference Laboratory, so we get a lot of information from people that we work with. So when there's been outbreaks in the past, we've been able to gather data. It's not a perfect system by any means, but it does allow you to get some intelligence with regards to what's going on the ground.
Okay, that's good. I had a personal question, but conscious that I did lose connection for a little while, so maybe you answered it. When you were talking about the emergence of PNW variant that is highly pathogen, you spoke about outbreak in 2012 and 2013, but nothing subsequently. Have you been able to link these outbreaks specifically to heat waves, for example, or not the heat waves, but water temperature changes?
Yeah, so the outbreak in Spain in 2012, that was linked to very warm weather conditions in northwest Spain. And I'm not absolutely sure what happened there, but that was some factor involved in what happened there. We have had other outbreaks associated with those strains in places like South America now. I think also in Australia and New Zealand as well. So it has moved, but actually, luckily, I don't think we've seen very much in Europe. We have had subsequent infections, subsequent cases, but they've been very rare. Now that could be because it's not causing infections, it's not causing disease at the moment, or it could be because there's deficiencies with the epidemiology, the reporting, the surveillance, or it could be a little bit of both. So it's a difficult one because we should have expected to have seen more cases associated with that clone, and we haven't. When it started in 2012, I was sure that we would soon see infections all across Europe, but that hasn't seemed to have been the case. So it's sort of watch this space really at the moment.
Thank you. You know, so harnessing what you can do with satellites and following temperature changes is quite amazing. I must say, it's very, very convincing and cool set of tools to use there. I don't think there is further questions at the moment. There might be some later. I know colleagues who are on leave at the moment will catch up with the recording, so we might pass on some questions to you at a later stage. But if nothing pops up anymore, I think we might be able to wrap up a little bit earlier. In that case, I'll just thank you so much again for taking the time to come and talk to us about this really, really interesting, and as you say, as something to keep in mind for the years coming. Nothing coming up. We'll see you in the backstage, and we'll just thank the people who joined us today, and see you in future seminars. Thank you.